Oh, look, I have people joining in. Beautiful. Mid all. Okay. Good stuff, guys. Yeah, go ahead and start that first quiz. Like any other quiz, it should be in the assignments tab on the Blackboard. I keep wanting to say Slate. That's when we use it. Sheridan. It's on the Blackboard. It's online. You can probably do it on your phone if you don't have a laptop. Yeah, go for it. I think 35 minutes. I extended it to 35. Those of you who are online, raise inside. Sure. Those of you that are online, can you tell me if you can hear me? Just say like, I can hear you. Because I don't know where the mic is on this podium, but apparently I am making noises. Okay, great. All right. Now, do you get access to all these fun cameras? Oh, I think I'm using that camera right now. That's pretty cool. How do I switch cameras? Select the camera. Plus and just switch. That are online, start the online quiz. Right.
Just make sure they are you want answer for all of the questions, right?
I think so, yeah. Pretty sure. I don't think so. Is it? Oh, you know what? Some of them might have multi-select. Well, some of them might have a multi-select. Yeah. So
I can go back.
yes, some of them might have multi-select. Here, if you want to start over, I can make it so you have more than one submission. Okay.
I can
Yeah, if you want to,
because the reverse one, I can choose multiple
that's fine. Yeah, set it up so you have 2 attempts. Okay.
so I have to submit now and
Yeah, you can submit and do another attempt. That's fine. I'm gonna chalk this up to the first growing pains of running a quiz for the first time. Ready? Yeah, a bunch of them that have multiple. So. Yeah. You gotta go to the bathroom, or whatever? Go ahead. Yeah, we need permission to go to the bathroom. Okay, yeah. Open, what do you mean? Open book quiz? I would say no. Why? people online have big books open. Are they telling you about their open books? Or people have other tabs open? Yeah, so I had to come over and see your open tabs and give you guys zeros. Right, so this one should not require any extra notebooks, stuff like that. I hope you don't have any extra tabs open. If I had lockdown browser, I would just lock it. I can adjust these things. But on the Blackboard. I'm kind of working on like a hybrid of what the previous professor did and my own personal like preferences. So it's course information, value of your lectures lists. So he set it up so that quiz 1 is worth 10% of lecture component, 6% of the course grade. Quiz 2 is 8%. Midterm 20, quiz number 3 is 6, and the final 20.
I can leave, right?
What's that?
I can leave, right?
You go to the bathroom if you need to, or if you're leaving entirely, bye-bye. He's locked down, brother. Oh. Just close Blackboard. I want to close Blackboard. Guys, what are you doing? What are we doing? Leave a note today to make for myself later. Note to you guys that you will need your laptops. Sort of just what that is. Okay. Where's the mic in here? Almost done. Who's still working on the quiz? Working, still working, still working, still working. You guys start at 8, it would be around 8:35. So that'll be the maximum time and then we'll finish off. Right. I should do the quiz at the end next time, so you guys can all filter out done. Don't have to wait. It's a lot of weeks off. Okay,
So for the question that have multiple answers, right? What if you choose like two right, but you forget, but you then choose one,
yeah, 50%.
50%?
What's the 25% per answer for the full
Okay.
mark? If there's 4 things worth whatever. Okay, all done? All done?
Make sure you don't mention about.
Oh, was there a question about HPLC in there?
Yeah,
Oh, okay.
if it is vertical.
Or,
horizontal. I don't know.
Oh,
I
we
don't have
did.
any ideas.
We did talk about that. So HPL, so your classic column, who's still working on quiz?
It's a classic,
He's still working. Give him a second.
the same.
This young gentleman is still working on his quiz.
Same.
Yeah, one sec, guys. Well, we can discuss these things, no problem. Some people are still working, and some of my online friends might still be working. So definitely talk about it, sure. It's really awkward being on flex delivery, because it's like, what if someone online is like, I don't know if you can talk about it,
Live CM.
and then I'll,
So.
you know. Kind of unfair, it's almost like an advantage doing it at home, because it's like. I also, in the announcement, I said I want people to put their webcams on, and nobody's putting their webcams on. So like, how do I enforce this, you know? It's almost like I should make everybody come to campus for tests, do the class together. Right? I'm gonna talk to the other professors and see.
Where does that happen there? Keep shaking.
Email. Growing pains, guys, growing pains.
By this instrument, you can.
Where'd someone else get it?
Exactly. Not much.
Seneca. Almost done. And that camera, what about the other cameras? Okay.
I understand this. But you have to.
All good? Just two questions. Two questions. Okay. Must give. Okay. Okay.
I know, it seems like you can literally just. I would.
Let's go.
I mean, I didn't really. Depends on how. Yeah, I guess. I would be.
Everyone.
Take care, talking for one.
Start the 36, I believe, or 36. I think.
Everybody else still working? Okay, cool. All right, very nice. So yeah. All right, team. So let us, oh yeah, so I want to talk to you guys about the schedule here. The schedule I find is actually like really chill. Like you guys have a lot of days off. But I want to ask you guys. So it says week 5 test, no lecture. That's not our test though, right? Is that another test you guys have on week 5?
What?
I'm just saying because the test we're doing is on October 14th, right? For our test, our class specific, right? So are they doing this for a test you guys have in a different subject, or is this us in this class? All right, so I was wondering if you guys knew. Is this a question I need to ask? Because they just, huh?
October 14th is Thanksgiving, do we?
Oh, sorry, it's October 15th, the day after Thanksgiving. My apologies. Yeah, so the week of October 14th to 18th is our first midterm. All right, so there won't be a lecture that day, right? So there'd be a midterm on that day. So midterm, October 15th. All right. The quizzes will kind of work around it. Like this week, oopsies. The quiz, you know, this was supposed to be last week, but because I wasn't here, we didn't have it. And then the last test is on December 3rd. So it's midterm 2. or the final, I guess you could say. The final is on December 3rd. So we couldn't do our final in the last week, which I would have preferred to do, right? Because it would have given you more time for, studying, review and all that good stuff. But I'll show you guys this. This is the schedule of tests for other courses. So we had to make sure that things did not overlap. So those are our 2 tests.
I have a question.
Yeah.
Is a test for the final exam, based on topics before the,
You did cumulative.
yeah,
I always get that word, cumulative. Let me see, yes. Well, let me see. So final test based on week 6 to 14. So I'll keep it based on the stuff they just haven't been tested on yet.
so after the midterm test.
Yeah, so anything that hasn't been tested on yet will be in the final test. All right,
Thank you.
obviously there will be common sense things you will have to know that will travel through. You know what I mean? Like, what is chromatography? It's like stuff like that. All right, that will have to travel through with you, okay? But yeah, we'll keep it,
Like biology.
okay, so. Let's jump to what we're talking about in lecture today. All right, so it's actually really nice doing it on a computer like this because I can like 2 different screens. Okay, so what I want to go over today is we're going to go over this set of slides. We're going to go over, when's your guys' lab? Your lab's tomorrow, right? Your analytical lab or
Yeah.
tomorrow. So we'll go over what you're doing day 2 of experiment 1 tomorrow, right? So we'll go over what you guys are gonna do, what you should expect, and we can go over kind of the discussion questions at the end. You guys can, have a kind of preamble in advance. Amanda wanted me to go over the calculations. We don't really have any that I can see. It's mostly just what's gonna come off first, and then day 3, I think, is when you start doing calculations, like what's the void volume? What's the reaction time of this one or that one or stuff like that. So I'm not gonna worry so much about that. And for classic chromatography, it's pretty hard to do things quantitatively. It's more like qualitative, right? You know what things are. Quantitative stuff is a little more difficult in classic column chromatography. It gets much easier with HPLC when you can directly replicate conditions and you can be more precise with things, right? So other types of chromatography. So we have briefly touched on these ones. So we talked about chromatography based on polarity. We emphasized more on normal phase chromatography. Now we're going to do a little more of a deep dive into size exclusion and ion exchange. The stuff we're going to talk about, size exclusion experiments that I've never actually seen before, but are super interesting. The ones like an application you never really would have thought of before. Oh, I need to project, I need to share this with you, my friends online. Sorry guys, 1 sec here. Let's share that one. There we go. That's better. I'm going to turn off my webcam because I'm just showing students in the front row. I'm not showing myself. We're sharing, right? We're sharing. We are sharing. Okay. All right. Beautiful. Okay. So to remember, I have friends online and you see things. Would you let in Jenny? Okay, there you go. You're in, Jenny. Oh man, this is so weird for me. Okay, beautiful. All right, so yeah, we're talking about size exclusion, ion exchange, and the basics of spectrophotometry and detection and chromatography. These tend to go together kind of in a row, because when you do chromatography, you will tend to do a detection afterwards with spectrophotometry, right? So it kind of goes hand in hand. Okay, so size exclusion, as we've briefly touched on before, right, can also be called your gel permeation, your gel filtration, or your molecular sieve. All fun ways to sound extra scientific about, oh, it's a molecular sieve. So you can have different substances. You can have your dextran, which is Cefidex, which you guys will use in biochem. We're going to do a size exclusion for that for different proteins. You can have agarose, you can have polyacrylamide or polystyrene. Those are really fun. And so it separates things based on molecular size. So your larger molecules will move faster, right? Because they're unable to get caught in the pores. The pores are what slow the things down. So yes, it's kind of interesting to think that the smaller molecules will stay the longest, right? If you ever think about like you just have like a jar of like sand or something like that, small stuff always filters down to the bottom, big stuff stays on top, but this is the opposite, right? So it kind of is counterintuitive in some ways. So your macromolecules go into the pores and they will end up in the void, right? So what you generally do is, like I said last time, you'll have a marker molecule to mark your void volume, which is generally a really, really big molecule. You can use polystyrene, you can use blue dextran, is what they use in the biochem lab, because it gives you a nice blue color you can follow through the gel. So you can see exactly when things will come off. The one I used in shared in last semester for this was called this polyvinyl pyrrolidine or something, 3 million molecular weight, something like that. But I can only detect it with UV, so it became a big pain in the butt to get the void volume. All right, so what are the applications? You can desalt macromolecules. So a desalt solution, you can remove salt or lower the salt content of a solution if you wish from macromolecules. You can do buffer exchange, and you can do estimation of molecular mass. I believe you guys will likely do at least a little bit of estimation of molecular mass. There is a formula you can use for void volume versus the final volume of the smallest molecule coming off. That gives you kind of a sliding scale of how big something is based on when it came out of your sieve. So those are our 3 applications. Desalting, meaning removing salt. So you have a solution of, let's say, a protein. There's a really high concentration of the sodium chloride, potassium chloride, something in the solution. If you put that on a size exclusion column, the large molecules will travel through the column and the salt will stay behind because it's small. So you can remove salt from something without having to do stuff like, so we're looking for, so reverse osmosis or stuff like that. You don't have to be aggressive in your removal of the salt. You can just use a molecular sieve like this. So yes. All right, so yes, here's our example. So this is an example of a buffer exchange or a desalting. So hemoglobin, which has a molecular weight of 64,000. So you'd have to make sure that your gel has an exclusion limit of higher than 64,000. So that it actually travels sometime through the gel. If your exclusion limit is lower than 64,000, the hemoglobin will come off the gel really fast and you won't have any kind of travel, right? This is a really interesting experiment. What they do is, this is just desalting, but okay, so yeah, so I'm getting myself ahead of myself. So this is the desalting. You're transferring hemoglobin from a high salt to a low salt buffer. So here is the example of what they said here. So here's your column and your buffer. You add in your hemoglobin and your salt. When your sample goes in, the salt occupies 1 mil. Hemoglobin has 3 mils, but you'll notice the salt will start to travel at a slower rate. Hemoglobin, and it will come off the column first, and then you can get the hemoglobin without all of the salt. There it is. So in this case, again, this is only like finding out through experimentation, but you'll find that the hemoglobin can travel 3 milliliters every time you add a milliliter of eluent, or at least moves it by 3 centimeters, something like that. So it moves a little bit faster. Again, there's always eluent in the column, but we're adding our solution in there, and the main eluent in the column is a low salt buffer. Right? So there it is moving through, and you have your hemoglobin coming off by itself. So you just keep adding more mobile phase, and the salt will travel slow. And then there was the other one where you can oxidize or reduce hemoglobin as well. I don't understand the purpose of this experiment, except for just to see it change color as it comes through and kind of see the practical application of size exclusion. So you have your column separated with bands of oxidant, peroxide, and your reducer, which is called dithionite. So what you can do is you can put these 2 different things in the column, and you can have the hemoglobin travel really fast through these bands, and you can see it change color from red to blue to red, based on how it is oxidized or reduced by our oxidant or our dithionite. I like to have the visual, and this is the visual that the previous professor drew. And for those of you that know, I don't think blood changes to blue when it's reduced like that. I don't think so, but it changes slightly in color. I think it's a little bit darker. I don't think it turns actually blue, but I'd be interested to see if it actually does. I think the blue in your veins is actually just like an artifact of the color of your skin coming through, not so much the actual color of the blood. So you put the hemoglobin on top, you have these two bands. Yeah.
Break the red blood cell first, or it's
I'm
just
not
like a
sure.
blood?
I'm not
It's
sure
a
where they're
sample of
getting.
blood?
I'm not sure where they would get the hemoglobin from. I'm assuming that it's just like a pure source of hemoglobin they got from somewhere. The original source of hemoglobin, I'm not sure where they'd get it from.
I'm talking about molecule, not the cell.
No. Well, so anytime you want to do something like this, you would need to extract it from whatever source you have. Because if you have red blood cells, you have all sorts of stuff that would clog the column. It would be fun to be able to throw a sample of blood on here and watch it come through, but I think you'd have to extract it first. Yeah, that's a good question. And then, your protein extraction becomes a whole other fun experiment you can do. Right? But let's say we have a pure source of hemoglobin, throw it through the column. Our reducer and our peroxide are going to stay very long on the column. I think they will travel at some speed, but the hemoglobin will go faster. So it will travel through. As it passes through the reducer, it'll be reduced, become deoxidized, and you will get a blue color. So the non-oxygen attached version of blood that would be in your veins on the way back to your heart. And then it passes through another oxidizer and turns red again. Now, I would not expect it to actually be blue, but it's an interesting experiment. So I was looking up, I'd never seen this experiment before, and I wanted to just like see what the point of this was, like why we would want to do that. So I think it's meant to highlight redox reactions. So you guys can understand how redox reactions work and how hemoglobin works. And it gives you a nice visualization of the elution so that you see the change in the elution process as it comes off the column. And it can let you have some practical study of proteins and interactions with various chemicals. But as far as like what you might use this in industry for, I can't really see a purpose for messing with hemoglobin. Yeah, I like to see everything through like a practical lens. Like, why would I care about being able to do this? I can't really understand other than to understand how proteins work for something that's very interesting to see and to understand an oxidation reduction of proteins. But very interesting nonetheless. And it's interesting to see kind of the mindset of the previous professor and like what he actually cared about. This is interesting. Here's this one example. Now we're moving on to something else. So yeah, size exclusion is interesting. Practically, the way we use it for in biochem is to separate different size proteins. So if you have a sample of proteins that are mixed together, you can separate them. You can estimate their size, especially if you're trying to figure out what proteins are present. You can usually estimate, if you know the size, it gives you a clue of what protein it is. Right? You only get an approximate size, but if you know what the size is, you can sometimes figure out what it might be in a whole group of proteins. Okay, ion exchange. Okay, so ion exchange, very useful for dealing with analytes that have a charge. So the one experiment I did was with the amino acids. Right? So you can separate large and small molecules. If you can find a way to make their charge different, you can find a way to separate them using ion exchange. So examples, your acids, your bases, and your proteins. And by way of proteins, also amino acids, which make up proteins. Okay. You guys know what an isoelectric point is? Isoelectric point. So an isoelectric point in your amino acid or your protein, there's going to be a point where if you're a low pH, you're usually positive, and if you're a high pH, you're usually negative. At some point, when you increase the pH to a certain area, you're going to have an equal number of positive charges, equal number of negative, and so it'll be overall neutral, and it'll come off whatever column you have. So if you have a positively charged column or a negatively charged column, if you get to the isoelectric point, it will come off the column. You'd usually want to go above a little bit. So if you have a positive column, you want to try and get your amino acid, to be positive as well, so it gets rejected off the column, right? Or vice versa. So isoelectric point is the point you're trying to get above, or to, or above, or below, depending on your column, so you can get whatever it is off the column. That make sense? Yes, no, maybe so? Have you guys studied amino acids yet? In organic chem? No? Okay, so we can look up some amino acids if you guys want. I'm just looking, trying to make sure oh no, some people got, I wish it would admit people automatically. Sorry, guys. Everyone's like, sir, let me in. I'm like, oh, I'm sorry. Okay. Let's just look up some amino acids, just so you guys have some context. Amino acid structures. There we go. Can I pull this paper? Open image, a new tab. Sure, there we go. So for amino acids, here are your different amino acids. Right, so there are many different amino acids, right? There's the ones that we really truly care about are the ones that our body cannot manufacture, of which there are 20. So you need to get them in your diet, right? There's the chemistry gets really confusing because these can also be mirrored, but we can't use the mirrored version. So all we really care about are these forms of these amino acids. But you guys are doing organic this semester, right? Yeah, so all the different groups, all the different functional groups on your amino acids will give you different charges at different times, right? So at a certain pH, right? Oh, I can zoom in a little bit. At a certain pH, right, you will deprotonate this carboxyl group, and you'll have a negatively charged spot on the molecule, right? So all over the place, you have different functional groups. You have different spots at which you'll lose that hydrogen, and you'll have that negatively charged oxygen exposed. It'll then have a negative charge overall. You get a different point in the experiment when it will be neutral, you know.
You mean the acidic amino acid?
Yes.
The acidity or?
Yes.
Base.
Yeah, so that's why it's called an amino acid, because they all have these carboxylic acid groups that give off the hydrogen, giving out hydrogen into the solution, making it an acid, right? So if you know your amino acids very well, and again, to this day, I still have to look things up when dealing with amino acids. It never sticks in my head ever. I can never get it memorized, right? But you'll notice based on the structure of the amino acid, they will have different charges at different pHs. Because as you force protons to be removed by increasing the pH, right? You force the protons off, it'll be a negatively charged chemical, right? The lower you go down, the hydrogens get stuck to the molecule, it's more positively charged. All right, team? So yeah, just as like a background information. Where was I? Where was I? Talk about that more later. So yes, for a net charge. All right, so your stationary phase will generally be your ion exchange resin, which will have acid groups or basic groups that are immobilized. So if you have an acid group that is immobilized on the column, it will be a cation exchanger. So it'll be a negatively charged column. And so it will exchange positive ions. So positive ions will stick to the resin much easier. If you have basic groups, you'll have an anion exchanger. So it'll be a positively charged column. So negatively charged molecules will stick to it much easier and be retained for longer. The more your positive charge, the longer it sticks, right? And vice versa. So the slowing down, and then he needs to change his wording. The slowing down of analytes is brought about by the association of the analyte with the charged stationary phase through electrostatic attraction. Right, so opposites attract in this case, right? Positive and negative, right? It's kind of a little different than the other chromatography where polar, right, and polar will stick together. In this case, it's positive and negative will stick together, right? Opposites attract. Are we okay so far? Any questions from people online? All right. Okay, where were we? So, yes, so we need to make sure that our resin and our analyte have opposite charges. Otherwise, they're just off the column and you won't get any retention, right? So if you have any neutral molecules, you won't get any retention as well. So it needs to be the opposite charge to stick, or at least not neutral, right? So your net charges depend on pH in a lot of different cases. Some strong acids, like really strong acid resins, it doesn't matter what the pH is, they're always going to be negatively charged. This refers to its, I think it's elution capacity or retention capacity, which is in further slides. So yes, if you change your pH, right, and you can shift your equilibrium, right? So if you want something to stick to the column and they have different isoelectric points, you can make sure that your column is charged to attract one of them, and then the other one will come off, and then you change the pH and the next one comes off. So it's really helpful because there isn't really a speed at which they come off at separate times. You can dictate when they come off based on adjusting your pH. So in the amino acid lab in biochemistry, you guys will have buffers of pH 4, 7, and 11. So if you do pH 4, any amino acid that is, I think it said positively charged flammable. Yeah, that's positively charged at pH 4 will come off the column. Anybody who is still negatively charged, right, will stick to the column. Right? And as you continue to go up and increase the pH, they'll continue to get positively charged and they'll flow off the column. I can show you guys that as well. All right, so cation exchangers. These will have your negatively ionizable groups. So you can have strong ones, which can be like a sulfonic acid, which is ionized over the full pH range. And you can have weakly acidic ones that are ionized at basic pH. So this graph basically shows the exchange capacity. I find this graph really confusing, personally. But basically, if you have a straight line, it means that you have a good anion exchanger, but then it'll drop off as the pH goes down. So it won't be as good or as charged as the pH goes up. It just shows the exchange capacity. This is a graph I've never seen. It's a graph I don't expect to be very relevant. But just know that your strong acidic cation exchangers will be ionized over the full range. And if you're weak, then you will ionize at a basic pH. And I also wanted to look up what the practical application of having different, weak versus strong. So the reason you might pick a weak one, because why even bother with a weak one if you have a nice strong one? You might want a weak one so you can get different selectivity compared to strong. So you can get more nuance in your chain. So you can change the pH by smaller increments and get different things off instead of having to change it by like one full pH unit. More sensitive. You can do them under milder conditions. If you're really strong acid, you can't really handle very delicate biomolecules. So if you have a carboxylic acid one, you might be able to exchange things that are a little bit more delicate, require a bit more white glove handling. And they're a bit cheaper, the weak ones. So there's your practical application for those. Anybody else? Megan and she got logged out. Okay, we're still okay. If people online have questions, make sure you put your hand up or something so I can see. Okay. And then of course your anion exchanges will be your basic ones, which are your strongly one. Your strong ones will be the quaternary ammonium salts. So stuff like ammonium hydroxide. And your amines can be your weak basic ones. All right, so I would just know what the strong ones are and what the weak ones are. So an amine. Have you guys talked about amines yet in organic? Yeah, okay. So an amine, anything that has a nitrogen in there, in the chain, will be an amine. And it's funny, in high school, I missed the class for amines and amides. Yes. Yes. So your amide, excuse me, is when you have the nitrogen in the chain, I believe. And your amine is when you have an amino group, so NH3 or NH2. Right? Yes. So I missed the day in high school when we talked about that, and it's never stuck in my brain as well as I wish it would. So come to class. Okay, lovely. All right. So a preparative experiment. When you guys are preparing for an anion exchange experiment, and you guys might do a project like this. Do you guys have any project courses later on in your careers? Because I know at Sheridan, we have a course called, we have the chemical project course. We also have what's called a tech report. So students will make their own experiment. They'll do an industrial like chemistry experiment, right? And then there's a lower down version. It's the 2-year program, which is the one I run, which is the project course. I had them pick which column they wanted to do because we were going to do anion exchange. And then they had to figure out what buffers they needed to get things to come off the columns, right? So this is what you guys would do if you're preparing for anion exchange. You want to make sure you find a pH at which your column and the analyte carry opposite charges so that you can get them to bind to the column. If you have multiple analytes, then you want to make sure that you have multiple buffers so you can get things off the column at different pHs, right? So you want everything else that you don't want to hopefully come off the column without your, just your item of choice staying. And again, let me know if I'm going too fast, okay, guys? Too fast. Okay, so once we have things stuck to the column, we're going to figure out how we get our bound analyte off of the column. So we have to change our stationary phase, or if to get our analyte to be neutral. So you just switch to a different buffer. So you can have an example of a good buffer for this might be citric acid, because citrate has three different buffering regions. So you guys are able to make a buffer, right, at pH 4, pH 7, and then if you want to go even higher, you might have to do it different, right? But citrate's really good for those two different buffers. So you change your pH, throw in a different pH buffer, or you might have to change the ionic strength by adding salt to the phase. So if you throw a salt into your solution, it puts in positive and negative charges, right? So you have your positive, let's say sodium chloride, you throw in salt, you'll get positive sodium ions, and you'll get negative chlorine ions. If you throw that in, what'll happen is anything that's stuck to a negative charge will get competed by the sodium ions, and then vice versa. So if you have a positively charged column, the chlorine ions will then compete with the charges. And so things will get stuck to the column, and then anything else will flow off. So if you put enough salt on the column, you can wash your sample off as well because of those charges.
Take the salt out after that.
Yeah, so you might have to do a desalting step or something like that, depending on what you're trying to do. You can also, it doesn't have to be very concentrated, as far as I know. And also it might not even bother, whatever your next step in your experiment is, it might not mess with it, right? So the salt might just be a spectator for the next step, depending on what you're trying to do. But that's a good question. Yeah, so there it is. We'll need a desalting step, depending on what you're trying to do. All right, so pair ion chromatography is something I just heard about yesterday. So this is in reverse phase chromatography. So this is just a way to get something off a reverse phase column. Essentially what you do is you add a molecule with the opposite charge of your analyte in your mobile phase, and they'll stick together and come off the column together. I'm not sure why this is even bothered to be thrown in here. It seems like a little very specialized piece of information we really probably wouldn't need to know. But it is very interesting. So it's kind of a cousin of ion exchange, I guess you could say. So you throw in something that will bind with your analyte on your reverse phase column and then come off the column. So I guess they would use this to slow down migration of charged analytes in reverse phase chromatography. Okay. Okay, so are we okay so far with ion exchange and size exclusion? I guess. Are there any questions so far? You can always talk about things later as well. All right. Let's move on to spec. Elements of spectrophotometry. Have you guys done any spectrophotometry yet? You're not. Yes, no?
Yeah.
Yeah, you guys use like a Gen 20, Gen 30.
That's 30.
Gen 30? Yeah, at Sheridan, we still have the old Gen 20s. And in the micro lab, we have specs that are from the 70s. So they were like old when I was born. And one of the lab techs hurt her hand trying to get the lamp out, right? Something to do with the way the switch is kind of plugged into it. And I said, why don't we get the one that was in the 80s? Maybe they fix this problem. They won't listen. Everyone's scared about the micro lab. They don't want to bring anything in there. Okay, so spectrophotometry. This is so we can characterize or quantify a substance based on its ability to absorb light. So spec shows up in a million different fields, right? Shows up in astronomy, shows up in biology, shows up in physics, right? Shows up in chemistry, all over the place. Very, very useful thing to have. Right? So your spectrum will be your light intensity against the wavelength. You have your peaks, of course. If it's in infrared, you'll have valleys, right? Now, you guys have seen this all before, right? Your wavelength goes from your violet all the way to your red. The wavelength will increase as you go, right? So energy is dependent on the wavelength and vice versa. So the smaller your wavelengths, the higher energy you have, right? So the more blue or violet it will be, and then as you go, you'll turn into the UV range, and then of course going to your X-rays and your gamma rays and all that stuff. And then your red will go all the way to your microwaves and your radio waves and all that good stuff. So we have our different versions of UV light as well. I think your hard UV, ones that are really damaging, more the UVC, UVB. And your wave number will be the reciprocal wave. So the elements of our spec. So when you're working with a spec, you'll have your light source. Depending on what you're using, you might have a visible lamp and a UV lamp, which I believe is the deuterium one. It'll go through a monochromator and the grading. So the grading essentially separates the wavelengths out, so you can get different ones at different times, right? You'll pick the ones you want, and it'll go through your reference cell and your sample cell. So this is a dual beam spectrometer. So it'll compare the reference cell to the sample cell and subtract and give you the absorbance based on the subtraction of the reference from the sample. So if you go into the lab and you see the ones with the 2 spots for a cuvette, that's a double beam. And usually the one at the top is the reference, the one at the bottom is the sample. And you have your detector at the end. So yes, I believe your visible one is your tungsten and your UV is your deuterium lamp. So the monochromator, which is made up of your refraction, your prism, and your filters, is all in the purpose of picking a wavelength you want to shine through your sample. This is really important because if you want to get an absorbance at a specific wavelength, you need to have this function to allow you to do it. So I believe this is the prism. This will be your grading. I think the grading is literally just a mirror that is angled a little, like has little slices built into it to reflect different wavelengths at different times. The prism, I believe, is the one up top here. So this guy here. That's your prism. So the prism will split the light into its different wavelengths and reflect it onto the grating. And so the grating is spaced out in such a way that different wavelengths will reflect at different angles. And so you're able to pick one by angling this mirror, I believe, to pick a wavelength that you want. Again, the intricate details are, they're even like proprietary sometimes, and you can getting a nice, detailed diagram is hard of different ones. Which one? This one? Which one? That is it's a mirror that reflects in both directions. It's like half see-through and half not. It's like a one-way mirror that lets it bounce off in 2 different directions. So those are present in a lot of optical instruments, microscopes as well. So you'll have a mirror it's like a one-way mirror in one direction. And so it'll bounce off and go 2 different ways.
Oh, in the blank tube?
Yes, the blank, yes. So every time you have a mirror like this, for your guys' information, it takes away some of the light from your sample, right? That's why you need your photomultiplier. Every time you divide the energy, you're losing some of it, right? So single beams, I mean, I can't guarantee this, but a single beam probably needs like a less strong photomultiplier because it doesn't need to go through this whole bouncing around thing. So if you guys ever have a microscope that has 2 eyes and then one for a camera, you'll find it's a little dimmer than if it didn't have the spot for the camera because it didn't have to split into 3 ways. Okay, where were we? So, yes. So here's our sample compartment. The chopper is what I was talking about. Has partially see-through mirror to allow it to bounce in 2 different ways. Yes, your reference cell will be your blank, and your sample will be your sample that you're testing. So you can see why it's very important to make sure your reference cell is actually a blank and not a sample, because if you subtract a sample from a sample, then you've got nothing, right? So we measure them simultaneously and we subtract the blank signal from the sample so that you don't have to do it manually yourself later. So if we have a single beam, we have to run the blank and the sample separately. And that way the machine will have to do a subtraction, or you have to make sure it does, and then, or if it doesn't do it, you have to do it yourself. Now, in this case, I think this one, the chopper they're using might be a rotating mirror that bounces it in 2 different ways. So maybe it doesn't have a see-through mirror in this case. Maybe it's just bouncing back and forth and giving you kind of a blinking version of it. So it would work either way with the see-through mirror or with the spinning one. So if one blink goes this way, and then when it turns, it goes the other way. And so it just adds them up as it goes. Okay. So our multiplier, our photo multiplier, means the signal is going to be really short, right? It's going to be a very dim light. So it needs to multiply it up so you can actually get a signal, right? It's like an amplifier for the signal. That's all it means, photo multiplier. When you do a scan, when you're swept sequentially, oh my gosh, sequentially. Have you guys ever used the carry UV-Vis ones in the lab yet? So they're one of the functions, if you go into the folder, will be the scan function. So you basically run it from the lowest UV you can do to the highest visible. And so it runs them all through and then it'll give you an absorbance at whatever the absorbance maximum is. So that's what it means by a sequential sweep. So you kind of choose your wavelength and it'll just sweep all the way down and tell you which one has the best absorbance time. If you have a diode, you can detect them all simultaneously. A diode means you have a big chip. Each spot on the chip has its own spot for a wavelength detection. And so all of them can shine on it at once. And you're able to get every signal you want at the same time. So if you guys ever use an ICP, they have that same thing. They can measure like 60 different wavelengths at the same time. The one we have in the lab here. We got a brand new one at Sheridan that can do 5,000. Meaning it's got a chip that has enough little spots on it to measure 5,000 different wavelengths at the same time. So instead of where is it here? Instead of the light here at the grating having to be split or picked off, you can literally just do it all at once. I'm not sure how the structure would change a little bit for that one, for the diode, but that's how it would work. Okay. Yeah, so there's the diode. Okay, there it is. Yeah. So this is a very, very basic description, right? So you have your light source. Just making sure that people are all in if they want to be in. Have your light source going through your sample, bouncing off a mirror. It's going to go off another diffraction grating, which will split it into different wavelengths. All the wavelengths will bounce off the mirror, and then a spot on the diode will be ready for every single wavelength you're looking for. So you guys can imagine how precise the manufacturing has to be and how well calibrated it has to be to catch everything. Because if you go off by like, a micrometer, you're going to have really weird results, right? So very, very advanced, very interesting. And it also has no moving parts and can be very fast. Okay, so our spec, you can have two different ways. You can do absorption or transmission. And if you're doing fluorescence, you can do absorption and transmission, right? Or emission, I guess you could say. So you run your light through your sample. The sample will absorb some of that light at whatever wavelength you're running through, right? So if you guys see green leaves, right? They absorb every light except for green, and the green light comes back at you, right? So that's why they appear to be green, right? So the absorption is in the rest of the spectrum. So they drink in all the other kinds of light. That's why if you guys are ever in like an indoor greenhouse, they'll usually shine like blue light on them because that's the wavelength they seem to want the most, the plants, right? So you have your transmittance will come through. Whatever comes through is whatever your photomultiplier will multiply and tell you what's there, right? But at different wavelengths, this will change, right? So some things are clear at different wavelengths, and then like a piece of glass tends to be clear at visible light, but then when you put it in infrared, it blocks it. So if you were to look at it through an infrared lens, you wouldn't see anything through the glass. Does that make sense? So if you guys are using the specs, you have to use quartz as your cuvettes, not glass or plastic, because quartz is invisible to UV light as well, and also to infrared. So the way you can calculate your transmittance is you take your transmitted light and divide it by your incident light, the light you're putting in versus the light you're getting out. So for your infrared, this gives you a percentage. You can, anytime you're using a spec, if you guys are using the Gen 30s, you can change from absorbance to transmittance, right? So if you want to change it, doesn't really matter, it's just a button switch. So absorbance is related to transmittance as a negative log of that. So it'd be a negative log of the incident over the transmitted over the incident. So, and these are not absolutely true statements, right? Because you can use whatever you want for whatever application you want. You're not stuck to absorbance or transmittance, but they have their own, they have applications that are useful, right? And yes, your blank will be the IO, so your incident light. Right, because you're gonna have things in your sample that absorb, you wanna have a blank. Okay. So how can we apply it? So we can do a qualitative, which is what you guys will be doing in the lab, right? Because you guys are going to be doing in your analytical lab this week, you're going to be doing the actual chromatography, pulling the pigments off of your column. And then next week, you guys are going to be running them on the spec, as far as I can see in the manual. So you guys will see as things come off, right? You put your sample through, you'll get an absorbance for your chlorophyll A in different wavelengths, and you'll get your chlorophyll B. And this is why they appear to be different colors. So you have the orange one coming off the column, and then again, that's in the orange wavelength light that it's giving off, right? And then the green ones will give off in the green light, right? All right, so if we want to do quantitative analysis, we need to do the absorbance concentration relationship, which is based on Beer's law. So your absorbance is equal to the absorption coefficient. The absorption coefficient, you get it just through experimentation. So every substance will have its own absorption coefficient. You just have to look it up. Usually they'll tell you, right? Then you have your cuvette thickness. They usually leave it at 1 centimeter, so you don't have to mess with anything. You just leave it as absorption times concentration versus the optical path length. So it just ends up being 1 most of the time. So that's your Beer's law relationship. Basically what that means is your absorbance is related to the concentration, because the more molecules are in the way to block the light, the higher the concentration is. That means the more absorbance you will get. Very basic, right? Okay. So yes, when we use it, HPLC, you guys will be doing that based on Beer's law. So essentially what you have to do when you're doing this is you have to run some standards, right? You run the standards on your column and you will integrate the peaks. So what that means is you take the area under the peak of your standards, compare it, and you know the concentration of your standards. So if you compare the integration of the standard to your sample, you can determine what the concentration of your sample is based on integrating those peaks. So whenever you run an HPLC, you'll see under there, you'll see the retention time. The retention time will tell you what time your sample came off. So if you run your standards, your acetaminophen standard came off at this time, caffeine came off at this time, right? And then when you run your unknowns, you can say, okay, so the peak that came off at 1.2 has to be acetaminophen. And then if I area is the area under my peak, and I know the concentration is 100 millimolar, I can compare it to my sample, and I can do a bit of math, and I can get the concentration of my unknown. So it's really important to avoid kind of shoulders on the peaks, right? To avoid peaks overlapping, because the more you lose of that under area, the area under the peak, the harder it is to do your comparative analysis and figure out what the sample concentration is. So you gotta watch out for that. And we'll go more into HPLC in the next couple of weeks as well. So yes, the peak profile shows you the instant concentration of analyte in the eluent at the time. So your peak area is proportional to the original concentration in the injected sample. So this gives you your HPLC, lets you do quantitative analysis with the HPLC. And because HPLC is so, what's the word I'm looking for? Repeatable, you can make the conditions, you can use a computer to make it go the same thing over and over and over again. You're able to do the comparison with much better accuracy. Okay. So as Beer's law gives you your proportionality between your areas under your peaks and your sample concentrations. So this is how your absorbance is proportional to your number of molecules or your concentration. Oh, wow, that slide just ended. Boom, done. And we'll get more into HPLC next week. All right, so just to reiterate, you take you'll see this chromatogram come off, and the chromatogram will usually, now you call it a chromatogram when it's flowing off the column. So this you would call a chromatogram. So chromatogram, wink, wink, nudge, nudge. There's a question in the unit questions or the weeks questions about a chromatogram. On the x-axis, what do you guys see here? Minutes, right? So my column is flowing. As it flows off the column, I get different peaks coming off at different times. So on the x-axis is usually time. And then you'll have your unitless absorbance value or absorbance units on the right side. Right? So absorbance on the right, you can have transmittance if you want, but transmittance tends to be like a negative peak, I think, but it's not worried about that. So there's your chromatogram. When you're looking at a spectrum, so the spectrum being one of these, the x-axis will have wavelength or wave number and versus absorbance. So know the difference between a spectrum and a chromatogram. So this one isn't running over time. This one is just running over the spectrum. And then these ones are running over time. All right. So that is that one for this week. Things I need to clarify. Oh yeah, let's go to the lab manual here. Are there any questions so far? Right now? Okay, so let's go to the lab manual. Real quick, where did I put it? I put it here somewhere. This thing's in the way. It's not what I wanted. I wanted the lab manual. Where are you, lab manual? Here it is. Okay, so where were we? So this is what you guys are going to be doing this week. So day 2. So you guys have done your extraction and you guys have made your mobile phases for your, you guys have done this so far, right? Not just blowing smoke, right? Okay. So you guys have your mobile phases and you have your extraction. So what you guys are going to be doing is you guys are going to be packing a column. So you're going to set up your Illumina column. And you guys will notice you don't need, oh, you have, yes, yeah. So the way you do your column usually is you put your Illumina in the column. I think it has to be soaked in your mobile phase beforehand. So I think the Illumina you will be given will probably be in a liquid already. Not sure. Or you might just be soaking it. I know if you want to get really good results, you usually want to soak your packing material beforehand, but I'm not sure if you guys are doing that today. So you guys are going to be, yeah. Yes. You want your, so when you guys are packing a column, you do not want it to look like this. I wish I had, I need to hook up my laptop for the thing so I can draw. So here's your different parts of the column. You want to make sure that you're not getting a weird lump when you put the different things in the column. I think what you guys are doing is putting the Illumina in and then you're putting sand on top. The purpose of the sand is to make things kind of come down less aggressively onto the column so it doesn't sploosh your column material, makes it kind of soak down more evenly, right? So you want those to be straight lines so that everything goes into the column at the same time, right? Or at least in even phases instead of coming in at different moments, right? And that'll give you more pointy peaks, right? You want to make sure the water doesn't touch the Illumina. The water can make it clump and to make things not go through as easily. So you want to watch out for that. Let's not worry about that. So yes, 4 to 5 grams of Illumina. So I guess you guys are not putting wet Illumina in. You guys are using glass wool, sorry, cotton wool. So that's much safer than glass wool. I've never used cotton wool for this before. So I've used glass wool for these things before. It's literally wool made of glass. I find it kind of scary to handle because it's literally like powdered glass floating in the air, which terrifies me slightly. So you guys are using cotton wool, much better. Cotton might not hold up to certain chemicals, but for this one, it should be fine. And you guys are going to have your sand in the column as well. Have you guys ever set up a column before? I wish I'd brought one in for you guys, thinking about it. So you're probably going to be using a burette. You're going to pour your cotton wool at the bottom, right? If you don't put the wool in, then the Illumina will come out the bottom of the column and you won't get nice separation, right? So the cotton goes in the bottom. So cotton in the bottom, little cotton plug, then your Illumina, and then you're going to have your sand on top. Then you guys are going to fill it with hexane. So you're going to be dripping hexane through the column. Then you guys want to make sure that your flow rate is actually nice, because if your flow rate is too slow, you're going to be waiting 45 minutes for a drop and then you're going to hate your life, right? So this is the problem with column chromatography. It takes forever. But you guys are making nice fresh columns, so it shouldn't be clogged. It should, come off hopefully very quickly, but you also don't want it to come off too fast because then you won't get anything, right? It'll all come off at once and nothing will happen. So there's a nice balance you have to find. So I'm hoping that the 4 to 5 grams they give you will be a good column length, right? But yes. So make sure your column doesn't dry, because if you have a dry spot in the middle of your column, then nothing can get through. So you don't want that to happen. You also don't want any bubbles, because bubbles will mess with the travel of things through the column. You want it to be solid, no bubbles, no dry spots. And then that's what it should look like in the end. There it is. You want it to be a nice, short column if you can. You have your plug of wool at the bottom, and you have your sand on top. Now, I think there might be sand on the bottom as well. So maybe I missed that step. So I think plug of cotton, sand, alumina, and then more sand on top. Yeah, it looks like there's sand at the bottom there too. We okay so far? All right, so what you're doing this week in the lab. So you guys are gonna let your meniscus come to the top of the sand. Then you guys are gonna put your spinach in there. And you don't want to separate your spinach at all. You want it to all be on there at once, because you want it to all be mixed together so you can watch it separate as you go. And you guys are gonna put in your mobile phase, your first one, right? What's the polarity of the first mobile phase? High or low? It's gonna be non-polar, right? Hexane, non-polar. Then you guys are gonna increase the polarity as you go, right? So in sequence, you're gonna add all of your mobile phases as soon as it touches the top of the sand, because if you mix them together, your polarity will change. So make sure it's getting to the top of the sand, then you put the next one in. Otherwise, you might mix them up together. The sand is also really helpful because it'll allow you to not let the column go dry, the top of the sand go dry, then put the rest of the mobile phase in, and then you're not letting the column go dry, just the sand, which is totally fine. Because the sand lets everything through nice and quick to just come back together and make the whole thing liquid again, right? And you guys are gonna collect some fractions. So you guys will hopefully collect colored fractions separately. So I'm not sure what kind of tubes you guys will be using. Sometimes you collect in beakers, or sorry, in test tubes. Sometimes you collect in cuvettes for the Gen 30, depending on what you're trying to do. So if you collect directly into cuvettes, it lets you put them directly on the machine and measure them right away. The only problem is if you use different cuvettes for every sample, because they're not matched, you can never do quantitative analysis with this. Because if there's a little scratch on the cuvette, then it'll mess up the absorbance a little bit. So you won't be able to get things very precise if you were to actually try and measure using this technique. So the only thing you can do is qualitative. You can tell where the absorption peak is. But you can't tell how much you have. So you guys will learn the importance of matched cuvettes a lot. I've never seen it really become a problem personally, but you always want to make sure your cuvettes are in the right same orientation and it's the same one if you can help it, right? Or they have to be very, very close. Exactly. So you guys will expect your fractions to be different colors. You would expect your yellow to be first, then grayish green, then pale yellow, oopsies, then a deep green. That's what you expect. Now, expectations are one thing, and then reality is another. It might come off in a completely different way you weren't expecting. So it happens all the time. Roll with it and see what happens. The best thing about science is weird science. Right? Then you guys will go into day 3 and you guys will determine what exactly it is. So let's just really quickly talk about what you guys will expect to come off first. So I want to go through the structures here. So here's our different chemicals we expect to come off the column. Right? Yeah, beta carotene, xanthophyll, chlorophyll A, and chlorophyll B. So I think we might have talked about this before. Which one of these do you guys expect to come off first? What do you think?
Shoot, the first one.
Yeah. Which one is the least polar? Beta carotene. Yeah. Why?
They are just measure carbons.
Yes.
We have oxygen or nitrogen.
Yes. So
Function.
yeah. So it's a carbon and hydrogen, right? No really electronegative molecule or atoms on there, right? So no oxygens, no nitrogens, no phosphorus, no sulfur, right? So and the chain is generally symmetrical in both directions. So you don't have even van der Waals won't even give you much of a charge, right? So least polar. Then xanthophyll has one group on it, right? So that one will probably be next. Right, so again, it's asymmetrical. And also has the OH groups on it. As you go down to chlorophyll A, you've got 1, 2, 3, 4, 5, 6, 7, 8, right? Nitrogens combined with oxygens. And then, so I'm not sure which one comes first, A or B. Let's see. 1, 2, 3, 4, 5, 6, 7, 8, 9. You getting that right? So which one comes up first, A or B? A probably, right? So A has 8, I believe, if I'm counting correctly. And chlorophyll B has 9. So you expect that one to be last. So you can associate them with the colors, right? Lovely. Okay. Where is that set of questions? Where is it? Where are you, question list? All right, so these are ones you guys should be able to answer based on this week's stuff. Okay. All right. So the axis of a spectrum, and we won't go through all of them, right? But maybe we will. I don't know if we have enough time for all of them, but let's go through it. So the axes of a spectrum are the difference between spectrum and chromatogram, right? So what's on the x-axis of a spectrum? Wave, yes. What's on the axis? What's on the y-axis of the spectrum? Observance. What do I do with it? Here it is. Yes. Or magnitude of signal or intensity units. All sorts of different ones they use in different machines for measuring things. Okay, so chromatogram. What's on the x-axis of a chromatogram?
Units.
Units of time. All right, I don't know why I did that. And on the y-axis,
Observable.
the absorbance or the intensity units or the units of intensity, right? You can also have percent transmission, which is just the inverse, right? All right, which one's the highest or the increasing wavelength? Who has the lowest wavelength of those three? UV. And then it goes to,
Visible.
yes. So they're in the opposite direction, right? So it goes ultraviolet, visible, and then infrared. So we do have infrared spectrometers here at the school, right? They're very interesting because you usually have a valley instead of a peak. Not sure why we use valleys instead of peaks for infrared. I've never really asked the question before. But if you ever see an infrared spectrum, it's like a normal spectrum, but flipped over. All right, 254 nanometers. Now that was in the list of different wavelengths we went through, right? 254 is in what spectral range?
UV.
Ultraviolet, yeah. So anytime you see 200 something, it's probably UV, right? So you guys will find that really annoying sometimes if you want to use a certain chemical because it's nice and cheap for your experiment, but to detect it, you need a UV vis, it becomes really annoying. Because the easiest experiments are ones you can use a Gen 34 because they require less preparation, right? You don't need an actual giant machine. It's much cheaper to operate, cheaper to make, right? All right, so why does chlorophyll look green?
Doesn't absorb green.
Yeah, so absorb that wavelength that is not whatever it gives off, which is green. So you'll see it absorb in the red and the blue, basically. So if you looked back at the slide, you were in there. Where were we? I believe if you guys were to look up, where are we? If you guys were to go to this spectrum again. If we were to look up the wavelengths of the green one, I'm assuming it's green because it's green. If I looked at the wavelength here and the wavelength here, it would be a wavelength that probably isn't green. I'm assuming, right? Chlorophyll B can absorb in such a way that it gives off an orange. Right? I believe. Now, that's again if these colors are matching which one is which, right? Okay. Let's see. Pretty sure we didn't talk about quartz. I think quartz is UV as well. I'd have to check. Tungsten though, your tungsten bulbs in the ceiling are usually visible light. You give off kind of a full spectrum, right? So you get heat, you'll get the visible light. You won't get so much UV. You'll get a little bit from tungsten, I'm pretty sure. And you also get that far infrared, which apparently is really important for our health, right? So I'll have to look up the quartz. Well, actually, no, I have the answers here. I wrote down the answers myself here. Where are we? So quartz emits UV. So a quartz lamp will emit UV radiation, right? You guys know what gives off X-rays? I think it's is it nickel? It's a nickel isotope or no? Something. It's I'm trying to think of it right now. I'll tell you later. But yes, different ones give off. I think you need radioactive ones to give off X-rays and stuff like that. All right, so single beam versus double beam. What does single beam mean? We pick, well, you also have the sample and the blank are not measured at the same time. The single beam meaning we can only measure one thing at a time. We can't measure sample versus blank simultaneously. Right? So double beam means I can measure sample and blank simultaneously. All right. Does a single beam need a blank? Yes, you always need a blank. Right? What constitutes a blank? You guys done blanks yet? You accept?
Everything except the analytic.
Yes. Now there are different kinds of blanks. There is a calibration blank, which is usually just your diluent, your water, whatever acid you're using. There's a sample blank, which is a sample you've made in the exact same way, but that doesn't have any of your analytes. So those are difficult to make because sometimes the analyte is built into your extraction technique, right? So it's hard to do certain kinds of blanks. Okay, I don't want to dig through too many of these. Oh yeah, this is a good one. What are units of absorbance measured in? There are no units. Yes, it's almost like a relative thing, right? I have more absorbance than the blank, right? Okay, for people that one I'm not sure about. Let's see, I have the answer here off the top of my head. So the traditional limit is an absorbance of 1. Sure. That's where I got offline. Traditional limit is an absorbance of 1, but you guys will find that your absorbances will exceed 1 many times. So that question seems kind of silly to me. Kind of dumb. Let's keep going. Not do too many questions. Oh, okay, it's not that many. Let's do one more, maybe all for you guys. Here we go. What happens to an analyte carries the same charge as the charge of the ion exchange resin in ion exchange experiment? I have a positive column and I have a positive analyte. What will happen? So will it come off? It'll come off the column quick or slow?
Quick.
Quickly. Beautiful. Lovely. Okay. Good stuff. All right. So yes, be ready to answer those kinds of questions for our next quiz. Our next quiz. Let me see if I decide when we should do that. Because again, I'm still just running on what the previous guy did, right? So schedule that I posted on amino acid structure. That's not what I want. I'll make sure you guys are well notified of the next quiz. I think it's based on week 4 and 5, something like that, right? Or 3, 4, 5. Are there any questions? All right, I'm going to try and compile this Zoom meeting. My Zoom friends online, do you have any questions? All right, guys, have a lovely day. I'll see you next time. We'll clarify the test things. I'll clarify it and get back to you guys as soon as possible. Okay? So you know which weeks we have off, or at least which weeks we have a test. So, okay. Yeah.